Hi guys, this is A2 Chemistry, Organic Chemistry, and we are doing drug synthesis. Question 6. The nature and variety of drugs that are available to treat diseases or life-threatening conditions has never been greater. At the same time, we are much better able to deliver drugs to their targets in the body. Some drugs have to be given by injection rather than by mouth. Name a functional group in a drug molecule that might be broken down by the acid in the stomach. Acid hydrolysis of amides and esters can occur if the drug molecule contains any amide or ester bond. So, both of them are the functional groups in a drug molecule that might be broken down by the acid in the stomach. So, amide and ester are the functional groups. So, amide and ester. The anti-cancer drug Taxol could be broken down if taken by mouth, and the structure of Taxol is given. Circle two bonds, each in a different functional group that could be hydrolyzed in the digestive system. So we need to identify ester and amide bonds. So an amide bond is seen here in this circle. This can be broken down by digestive system, and we can see ester bond. So this is an ester bond here. This can be broken down. This is an ester bond here. This can be broken down, and another ester bond here. So there are three ester bonds present in this molecule, and we have a single amide bond present in the same molecule. So any one of the ester bonds and the amide bond will be a suitable functional group to be circled that can be hydrolyzed by digestive enzymes. Question 7. When liposomes are used to carry drugs, their main purpose is to prevent the drug molecule from being broken down on passage through the digestive system. Name a functional group present in drug molecules that might be broken down by the acid in the stomach. So, if we have an amide bond or an ester bond present in the drug molecule, these groups can be broken down by the acid in the stomach. Name the type of reaction that would cause such a breakdown. Since the acid is carrying out the breakdown, the type of reaction would be known as acid hydrolysis. The drug sirolimus is used to suppress possible rejection by the body after kidney transplants, and the structure is given. Circle two bonds, each in a different functional group that could be broken down in the digestive system. So we need to identify ester and amide bond. So this is an amide bond with the C double bond on next to a nitrogen atom, or like directly bonded to the nitrogen atom. So this is one of the functional groups that could be broken down. Another would be an ester group, which is found right besides the amide group. So this is C double bond O, single bond O. So these are the two different functional groups that could be broken down by the digestive system. Question 8. Many common drugs are taken orally, but some medications, such as those based on protein molecules, are injected to prevent them from being broken down in the digestive system. Name a functional group present in drug molecules that might be broken down by acid in the stomach. So these functional groups include ester and amide groups. State the type of reaction that would cause such a breakdown. Since the stomach acid is carrying out these reactions, so the type of reaction is called acid hydrolysis. Which one of the following compounds would not be suitable to be taken orally? So, out of these three compounds given, A, B, and C, we need to identify the compounds having amide and ester bonds. So, in compound A, as such, there are no ester amide bonds present. In compound B, there is an amide bond present here. an ester bond present here and an ester bond present here. So we've got one amide and two ester bonds in molecule B, and in molecule C, there is no ester or amide bond present. So out of these three, the one that would not be suitable to be taken orally is the one that contains the amide and the ester bonds, which is compound B. On the structure you have chosen, in 3, circle all the functional groups that might be broken down by the acid. So 2 ester and 1 amide groups have already been circled. Question 9. Oseltamivir is an antiviral drug that slows the spread of the influenza virus. The structure of the drug molecule is represented here. Circle two bonds, each in a different functional group that could be easily hydrolyzed in the body. So these would be ester and amide bonds. So an amide bond is found here in the blue circle, and an ester bond is found here in the red circle. So both type of functional groups have been identified in this drug molecule. Oseltamivir is a chiral drug. This drug is usually taken as a single optical isomer rather than as a mixture of isomers. Suggest one benefit of taking a drug in this way. So, if a drug molecule contains a chiral carbon atom, it means that only one of the two optical isomers is the one responsible for the beneficial effect. The other causes the harmful side effects. So, suggest one benefit of taking the drug in a single optical isomeric form: reduces side effects because the optical isomer that causes side effect is not taken. Another reason could be smaller quantities need to be consumed because only the isomer that is useful will be the one that is consumed. So half of the total quantity of the drug would not be required. And another reason could be if you take a single isomer, only the useful isomer when consumed will bring about positive activity. Since the harmful side effects are not there, the useful isomer when consumed alone will bring about improved activity when it is consumed. Question 10. Many drug molecules are chiral but are often produced as a mixture of optical isomers. Suggest why a larger mass of a mixture is required than a single optical isomer. Only one optical isomer brings about the therapeutic effect. In combination, double the quantity of the useful isomer will bring about the same effect. Because if we need, for example, 10 grams of the useful isomer, we'll need to consume 20 grams of the mixture in order for 10 grams out of that 20 grams to bring about the therapeutic effect. Suggests a problem that might arise as a result of taking a mixture of optical isomers. So only one of the isomers is responsible for the therapeutic effect. The other isomer may bring about the harmful side effects. So the useful isomer will bring about the therapeutic activity. The other isomer that is not bringing about the therapeutic activity will bring about the side effects. Question 11. Aspartame is an artificial sweetener that has the structure shown below. Draw a circle around each chiral center in aspartame. So we need to identify chiral centers here. So in order to find out the chiral centers, we need to look at the carbon atoms that are bonded to four different groups. So starting from left, the first one has been circled here because this is the carbon atom bonded to four different groups. Moving onwards, we have another carbon atom with attached to the NH2 group, which is bonded to four different groups, and till the end there are no others. So there are two chiral carbon atoms present in a molecule of aspartame. In the stomach, aspartame is hydrolyzed by acid to form three organic products. On the diagram above, use arrows to indicate the two bonds that would be hydrolyzed in the stomach. So the two bonds that would be hydrolyzed in the stomach would be the ester bond and the amide bond. So these are the two bonds that would undergo hydrolysis in the presence of stomach acid. Draw the structure of the three products formed after complete acid hydrolysis of aspartame. So if we begin from left to right, the first molecule would be CH3OH, which is methanol. The second molecule would be COOH. Then we would have CH, CH2. Then we would have the benzene ring above. And this continues to NH2. This will be the second molecule that will be formed upon hydrolysis. And the third one, which will be formed upon hydrolysis, would be COOH. Then we would have CH, NH2 here, CH2 in between, and then again COOH. This will be a dicarboxylic acid with an amino group on it. So these are the three products formed after complete hydrolysis of aspartame in the stomach. Aspartame is soluble in water. By referring to the structure of aspartame, explain why it is soluble in water. Presence of the NH, NH2 and COOH groups in aspartame results in the formation of hydrogen bonds with water, making aspartame soluble. So the NH group, the NH2 group, and the COOH groups that are present are capable of forming hydrogen bonds with water molecules, because of which aspartame is soluble in water. Question 12. Prodrugs are compounds that are inactive but are easily converted in the body to the active drug by enzyme hydrolysis. Compound W is a prodrug. Complete the molecular formula of W. The number of hydrogen atoms have been given, which is 34. So let's count the number of carbon atoms. So 1, 2, 3, 4, 5, 6, and 6, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, and 24. We've got 24 carbon atoms present in this molecule. The number of nitrogen atoms are 1 and 2, and number of oxygen atoms are 1, 2, and 3. So the molecular formula of W is C24H34 N2O3. Compound W contains a benzene ring in its structure. Yes. Name three other functional groups in W. So starting from left, we can see an amine group, the NH2 group. Then we have the NH2 group attached to an ester group. So we've got two amine groups, an ester group, and moving on till the end of the molecule, the last cyclic structure has a double bond O present on it, which is a ketone group. So the three functional groups present are amine, ester, and ketone. On the diagram above, use an arrow to indicate the bond that would be hydrolyzed. So the only bond present in this molecule is the ester bond that will undergo hydrolysis. This is the ester bond. Draw the structure of the likely products of the enzyme hydrolysis compound W. So the likely products for this would be, if we begin from the left side, H2N, then we have got this part with another NH2 here, and going down we've got C double bond O, and upon hydrolysis this will form OH. So this is the first product upon hydrolysis. The second product upon hydrolysis would be the oxygen will be attached to hydrogen, and this will form phenol. Then we have the remaining molecule bonded to this phenol molecule. This is the second molecule that would form. What features of the molecule W makes it water soluble? Explain your answer. Presence of NH2 groups results in hydrogen bond formation with water, and presence of COO groups result in ion dipole attraction with water, making W soluble in water. So presence of hydrogen bonding and presence of ion dipole attraction are the reasons for the solubility of W in water.